Hello, everybody. I'm Scott. I'm a computational biologist in Dr. Lindsay Cowell's lab. Our lab has three sort of main focus areas. We do method development, so new algorithms, analysis, lately sort of machine learning algorithms for immune repertoires. Our other area uh, is is resource development and deployment. And our third area is actual research projects, research on with adaptive immunity, primarily in the cancer space and in neurological diseases. Today, I'm going to talk about um, our resources that we've deployed, developed some existing ones as well as some new ones coming down the road. So about the first half of the talk, I'm going to talk about VDJServer, which is an analysis portal, and all of these resources have used the Immcantation tools to some degree. I'll also mention about the Air Data Commons. We've heard probably a couple times about the Air community. We're very much involved in that community, both in the standards and in some of the resources involved. And then lastly, I'll talk about a new resource which is coming that's recently been funded by NIAID called the Air Knowledge Commons. Okay, so what is VDJServer? Well, VDJServer is a analysis portal and data commons. It was funded by NIAID, back a long time ago. It's been publicly available since 2015. It has sort of three main parts to it. One is a data management infrastructure. Essentially allows you to load up your sequencing data, your immune repertoire sequencing data, keep it in sort of private project areas. You can share those with lab members, and like when you get to publication, you can make that data and analysis available. What we've been working quite a lot on in a few years is working closely with the Air community to follow the Air standard. So besides the say the sequencing data, you can also put in metadata about your subjects, your samples, and create groups for cohorts for comparative analysis and so forth. And that's all sort of based upon the Air standards. The other part is the analysis portal, sequence processing. This is where we have a set of pipelines. We run the analysis on a supercomputer that's provided by the Texas Advanced Supercomputing Center in Austin, Texas. That compute is free, so much of our focus is for researchers who maybe don't have the computational cluster available, or even sort of the bioinformatics skill. They can go to VDJServer through a web interface, upload their data, run it through sort of a standard pipeline, and we provide it all for free. And the last part is we have a number of sort of visualizations, so you can visualize the output of the analysis, narrative analysis, and so forth. So I'll show you some examples of that. So really roughly, the VDJServer, the architecture is sort of a standard, what I would call a two-tier architecture. The first is you use your web browser, and the application runs in the web browser. It then communicates to some server processes using APIs. And there's sort of two main ones there. There's what we call our VDJ API, which is has very specific functionality for VDJServer. And then we rely upon another set of APIs called TAPIS, and this is provided to us by the Texas Computing Center. It's a really nice framework because it handles a lot of the sort of complicated infrastructure things like running and managing jobs, managing users, interfacing with a database, and so forth. So as I said, VDJServer provides a number of analysis workflows. Now, our approach has been kind of to provide mostly roughly a single standard workflow. So that has those advantages and disadvantages, right? In that, if you need to do something sort of custom, that's, you might have to then, work on your own sort of workflow for that. But, in the most part, the standard one works for a lot of people. And then, you can get the output and you do some additional stuff. So there's a rough aspect of, what the input files are. This right now, this diagram I'm showing you is often is the workflow for bulk immune repertoire sequencing. So you often have paired end reads or some sort of sequence read files that you do some pre-processing on, quality control. Once you're fine with that, then you do VDJ assignment on that, and then finally there's a bunch of post analysis that you would do. So I mentioned now, if you have single cell, which is now becoming very common, this changes a little bit in that you typically, at least we're talking with 10x genomics, you're using Cell Ranger to do the initial parts of that, but then that data can then flow into the analysis routines, and you can use those just like you would the other bulk data. Okay, so as I mentioned, we use Immcantation quite extensively in our pipeline. These are the tools that we have available and integrated. So we have Presto available to do pre-processing. We have another tool that's our own for pre-processing that it has sort of limited capabilities, but both are available. Changeo for calling, for clustering and doing clonal assignment, and Alakazam and Shazam. So these are sort of the downstream ones. As I say, we have a number of other tools. I won't list them all out there, but I'm welcome to give you information about that if you're interested. Okay, so as I said, I think one of the really nice things about VDJServer is it provides a number of visualizations. So after you run the various tools or the pipelines, you can interactively use your web browser to look at these visualizations, make some sort of decisions. There's sort of two aspects about this. So the ones I'm showing here are pre-processing visualizations, right? So as I said, that first step is you go from your raw sequence files, you do some quality control checks and so forth. And so there's a number of these that you can then look at. So you can look at it a sample by sample basis, and determine if there's any samples that were not, you have some issues with the processing. Maybe you want to go back and change parameters, length cutoffs, quality cutoffs, and so forth. And these visualizations, what they're showing you, it may not be quite easy to see, is they're showing you a pre and a post. So they're showing you sort of before any filters are applied, and then after, so you can see the effects on these various metrics. And then our other visualizations all are sort of at the analysis stage. We have a number of the sort of standard, standard sort of figures and charts, like to look at the gene usage, length histograms for the CDR3 junction, clonal charts, diversity charts. We have this nice selection quantification, which is one of the calculations that comes out of the Immcantation toolkits, where you can look at selection pressure on the various regions in your V gene. These are all interactive. So I'm just showing you a static snapshot here, but all of these have drop down buttons where you can pick what samples you want to display. If you've defined groups, You can then show multiple groups on there. B here is an example. This is group, and I can tell that because it's showing you, it's actually averages with error bars for two group comparisons here. And so, as I say, so it's very interactive. You define all your groups ahead of time, and then the analysis will then provide you, and you can go back and forth. And then if you find, okay, well, sometimes, the visualizations are good, but you want to do something more custom. The data, typically they're in sort of spreadsheet format, either, TSV files or CSV files, so you can easily load them up into Excel or R or whatever tool that you'd like to do your own custom analysis with. What we've been doing lately is adding some additional sort of more sophisticated visualizations, and to do that we've added support for other plotting libraries. The ones that I showed you before, those are figures that are generated right in the browser dynamically as you select them. Some of these more sophisticated figures, we now have the ability to generate them on the server, also dynamically when you request them, but then display them in the browser. So here's one that I was working on for a B-cell study. It's showing you the mutation rates at each of the individual amino acids for a set of B cells. And then this other one over here, I apologize, the screenshot's not so great, but this is sort of a graph visualization with nodes and edges that is showing an analysis provenance. Okay, so that was VDJServer. I'm going to then talk now a couple of other resources, in particular the Air Data Commons. And as you heard before, and you probably already know about the Air community, which was formed a number of years ago, that was sort of working on developing standards for data sets, as well as things like file formats, so that analysis tools could all sort of interoperate much easier, because they can then, the outputs of one tool could be fed into other tools, and they can all sort of be used together. So I myself personally have been involved in many of the working groups, and so I'm familiar with and been involved in developing these standards, and I've been working to sort of include them in the VDJServer. One of the working groups, the Common Repository, was very interested in how can we share these AIR-seq data sets, not just the raw data, but the processed, analyzed data, so that it could be queried and downloaded, so people can do additional analysis. So that's really the basis of the Air Data Commons, and we defined an API for it. And we at VDJServer, we want a really large repository of it, but there's also other ones as well. And so Immcantation is sort of a critical part of all that. So a lot of the data in the Air Data Commons has been processed in sort of a standard way with using some of these Immcantation tools. And I think that's really good. I say here, it kind of flows through all of it, from the data entry all the way to where you publish your studies there. So I'm very happy because this data commons has grown quite significantly. We have well over 5 billion sequenced annotations in the data commons. The number of studies doesn't sound nearly impressive. It's around 90 studies, but some of those studies are very large, contains both B cell and T cell, and it's across a number of diseases. There is some mouse data, but the majority of it is human data. And so this is just showing you sort of the growth of it. And we sort of expect this to continue in the future, because there's many studies that could still be curated and put into the Air Data Commons. So it's, as I said, there's an API, which is sort of a programmatic thing for, programmers and bioinformaticians to use. In VDJServer, we developed a GUI interface that we call a community data portal. Now that actually has multiple parts. So if you can use VDJServer to publish your studies, so you can upload the data, put in the metadata, and then publish it, and then people can download it, query it, and also look at like analyses and visualizations that you've done. So as I say, we're one of the data repositories. We're pretty much the we're one of the largest ones. About half of that data is in VDJServer. I didn't mention it here. I should have added a bullet point that we are, for NIH data sets, we are what would be called the preferred data repository. So if you're somebody who's writing NIH grants, now that they're actually asking for data management plans, how are you going to, share and publish your data? VDJServer is a NIH approved repository for that. So if you're generating immune repertoire data, you can say, hey, we're going to put our data in VDJServer, and you now meet that NIH requirement. Here's an example. Here's just a screenshot of the community data portal. Sort of your standard sort of search. You can filter on specific fields if you want. And we have some statistics here. There's also some charts that are available that I'm not showing here, but I always like to show it's great how these numbers have really grown. So we almost, so we're getting close to 10,000 repertoires, say over 5 billion rearrangements across many repositories. There's bulk data. There's also many 10x single cell data is available. And it's very easy to download. You just click the button, and then you can download all the data for that study. And the data that you get is all in the AIR standards format. So those standard formats I've mentioned before, the data is all in that. So it's immediately ready to then plug into any sort of custom Immcantation analysis you want to do, interested in. Okay, so that was the Air Data Commons. Now I wanted to mention this last sort of new resource that's being developed, the Air Knowledge Commons, we're calling it. So right now today, if you're doing sort of a complicated adaptive immune study, there's typically multiple resources that you might want to utilize, pull data from, and so forth. So here's two. One is VDJBase, which has information about genotypes and haplotypes. OgreDB is part of the AIR community and is a resource that curates germline gene sets. So those the actual germline genes that you use for VDJ assignment. Some people use IMGT, but the AIR community has been sort of actively adding new alleles that are being discovered and creating curated sets. Another resource that you might use in your research is IADB, which has information on the epitope side, for both B cells and T cells. IDB is around for a long time, and there's a lot of great information there. You can search on, diseases and search for specific epitopes. They've been recently, over the past year or so, they've been, when they curate studies, they've been recording the receptors the immune receptors that are involved with that epitope. So now you have a little bit of that combined information. Okay. But the issue is, as I say, there's all these resources that if you want to pull the data together and answer a sort of a complicated question, and here's a use case, I'm not going to go through the details of it, but mention a different pieces of it. You know, if you're interested in a particular gene, in this case, this V461O2 allele that's involved in rheumatic heart disease, you might have a bunch of questions that you want to ask that cover these various resources. Well, today doing that is relatively complicated because you'd have to go each one separately, query it. Once you get the data, you download, you have to deal with all the different formats and how to merge it. It ends up being very complicated. And just to kind of give you an idea, like for this use case that we mentioned, this chart here shows how, you know, each of the resources have sort of different pieces of the total sort of pie that you might want to put together as part of your hypotheses and your questions. So so this has been around for a while, and so what did we say to NIDA? Say, well, hey, this is a critical need to really sort of bring together and unify these resources so that users kind of have one place to go, that they can do queries across all of that, get the data that's already in sort of a high quality, well curated format. And so here's just some information, right? So as I mentioned, here's sort of the different sort of repositories that are being integrated into this knowledge commons. There's sort of two big aspects of it. One is what you can see over here in this figure is what all these repositories do is they often get data from public archives, as well as user submissions, and those, you know, go through various annotation and validation processes before they go in the databases. So what we're working on is sort of a common data model that's going to unify all of that, the data across those different repositories. And then over in this other figure here, then data will become integrated. What I'm not showing in this figure is that there's going to be things where we add new knowledge as we do this integration. So we'll be doing sort of inferences about receptors and their epitopes and specificity and other types of things, and then making that available in a large knowledge base that you'll be able to query. We'll have a user interface that you can interact with. So I'm really excited. This is a project that I'm leading along with everybody on each of these repositories. They all have they're all part of this large grant, and so we're all working together to develop this. It just started, as I said, recently, and then we're hoping for a beta release. It's still a little ways away, about a year away. And that's what I have for today. Thank you very much, and I'll take any questions.
Thank you very much, Scott, for this talk. There's a couple of questions in the chat, but for the sake of following the schedule, maybe it would be great if you could answer them directly in the chat. Sure. So we will now move to the next speaker.